So, good afternoon. Just you have seen some active biomolecules. So, our next lecture will be very interesting. And Professor Anirban Sain will talk mechanics of shape change in soft and living matter. It is related to biophysics. And Professor Sain is working in the IIT Mumbai. He's a professor of physics. So that in the brief introduction, I'll request Professor Sain to start his lecture.
Okay, good afternoon. Session after lunch. Let's see if I can keep you awake. I have a worry that I have too many slides, but I don't have to really finish. So just ask as many questions as you want. I'll try my best. Okay, so this is a broad topic, and because Abhishek gave the morning talk, it made my job much easier because I'm going to talk about some active matter. And he tried to give you a fast introduction to active matter. I will not even attempt that. Rather, I will wave my hands and try to avoid equation and try to make it look possible. Okay, but keep asking. Okay, so what I am telling today is mostly about changing shapes of living matter. Okay, so by living matter, I specifically think of cells. Okay, so cell is a bag of fluid, very layman's term, and because it's living, it has to divide and it has to move. While doing these things, it has to change its shape. So a bag of fluid, a passive bag of fluid cannot change its shape. Something else has to be there, and this something else are these proteins which actually interact with this membrane. Membrane is the surrounding which confines the bag of fluid. And so these proteins interact with the membrane and somehow make things move. And while they move, shape also changes. Okay. So what I will try to motivate here is that how this protein membrane interaction leads to this change of shape and often makes things flow. Flow on the cell surface often. Okay, so let's see. So, but because we are physicists, we tend to simplify things. So one way of simplifying proteins is that thinking of them as rods. Okay. Of course, we know that they are complicated molecule, but they are not spherical. They often have one side longer than the other side with lots of branches, etc. But you can think of them as rod. Just like sometimes we think of elephants as points and make them do things what we want. Anyway, so proteins are rods for our modeling, and it does pretty well actually, meaning this simplification. Because there is a huge physics available, physics of rods, which are often called liquid crystal, very popular among chemists. It were very popular 30, 40 years back, but since last 20 years or so, liquid crystal physics, I mean, it's a mainstream subject in physics and it is finding a lot of application, especially in biophysics. And it's really rich. I'll try to at least show that. Okay. So, and the other thing that is special to biological systems is this activity, which is this last point. So activity essentially means that any living thing consumes food and it produces energy. Now, what does this energy do? Now, this energy essentially produce some work, which the cell does, and more microscopically, what it does is it generates forces. Okay, either forces, or if you want to think in terms of continuum, it generates stresses. Okay, so both these things, either you call it force or stress in a more continuum sense, they make things move. Okay, so activity is the reason for motion. Passive things don't move, or even they move randomly, but biological systems, because of activity, move in a concerted way. Okay, so source of activity is certainly chemical reactions. Although we will not, we don't have to think too much about that, I mean the chemical aspect of it, but we will be mostly confining ourselves to what they lead to mechanics. Okay, and I will try to impress upon you that in all this shape-changing business, mechanics is very important. Okay, of course, biochemistry is important. We don't really, physicists don't know so much of it, but mechanics is equally important. Okay, so before I forget, students at different times have worked on the things that I will be talking. So I will not particularly mention who has done what, but so this, I'm just showing the general names, and Suvechha is a current student. Okay, so so this is the example, just 2 examples I'm showing, the shape change of cells. Okay, so on the left, you see a movie, you will see a movie of cell division. Okay, so it's a cell confined in a shell, just like an egg, let's say. And you will see as I play the movie that it changes, it will divide. You can see what's happening. Eventually what is going to happen is at the middle, it is dividing. You don't see the wall coming up, but you see that it gets divided. Those are the 2 nuclei which has gone to 2 sides. But if you look a little more close using fluorescent microscope and so on, you will see what I am showing in the diagram below. That is these 2 lobes which are now 2 cells. They are dividing where I have put the arrow. And beneath the cell surface, which you don't see in the upper diagram, there is a network of filaments. They are actin. Okay. So now these filaments, it's like they are skeleton of the cell, and a lot of motion is actually taking place within this network of filaments. So it's like a sheet of filament just beneath the cell membrane, and it's responsible for changing shape. The second example, so I will be talking about this system quite a lot. Yeah, this is another example which we had worked on. Red blood cell. Everybody knows about red blood cell. Their normal shape is this biconcave, 2 dips in the middle, and a disc otherwise. So they often become anomalous because of sickling. This is the sickle structure. It happens because the hemoglobin that are there inside, they become fibers, and it's an anomalous disease condition. And these fibers are stiff, and hence they will deform this original cell shape. And because of that, these sickles find it very difficult to pass through capillaries, and they are very rigid actually. On the other hand, these biconcave shapes, they're pretty flexible. They can just squeeze through capillaries of all sizes. Okay, so we looked at why does this become like this sickle shape. So you just have to think of that. Suppose you put a stick inside a soft ball, a balloon, what will happen? It will deform that balloon. Okay, that's what is happening here. But this is one possibility that because so this is that stick I have put in. It's a flexible stick. It's not a rod. Okay, so it's a filament of hemoglobin, and it has certain bending rigidity. It's not like a thread which is flexible. It is not like a rod. It is somewhere in between. And there is some competition between this bag, which is made of membrane, the cell, and the rod, and they are adjusting to some shape which both of them are okay with. Okay, but of course there are other structures possible. Why does it go into this structure? It requires one to put in a little more physics into it, which I will not go into detail. But we had shown that if you have multiple of such filaments, actually it's known that sickling requires many filaments, not just one. This is just a physics model. Then you get complicated shapes. You can see here. And this is something which said new. That is that sometimes they can be like helical rods. I mean, the filament can become, so this is a hemoglobin filament. It can become helical. And because of that, you can see that it can deform like this. You can clearly make out that there is a helix inside this. This was there, but people never talked about it. So this is just an example how the bag of fluid, the cell, gets deformed because of somebody else. In this case, because of this filament network, this actin network just beneath, whose dynamics I will describe. And in this case, because of this fiber that is forming inside because of a disease condition. Okay, so these systems, these cells deforming, it's a composite system. It's not just bag of fluid, but there are other things, either a network of this actin or somebody else. So you have to be able to deal with both the membrane physics as well as the physics of filaments or the physics of network together. So this is a sort of a very typical thing in biophysics, that these objects like polymers, membranes, filament, etc., they are subjects of soft matter. Okay, you can separately study them. People do that, do experiments in vivo, mostly in vitro. But then when you come to the context of a real life living thing, you have to know how they talk together and give rise to something that you see. Okay. Sorry. Yeah. So now I am moving towards membrane, a little bit of membrane physics. So membrane is nothing but a sheet, okay, an elastic sheet. Now, membranes typically look like this. This is a cartoon. There are two layers. Okay, there is a head group, the yellow part, and there is a tail group, the tail staying inside. So this is a membrane, just a piece of a membrane. So membranes often deform because proteins, which is this green carved object here, and because this object is carved, it imposes its own curvature on the membrane, and hence the membrane bends. Okay, this membrane could be part of a cell. Now there are other proteins, membrane proteins, which are very asymmetric, which have asymmetric shape, a bigger head and a smaller tail, and they naturally tend to carve this membrane because they are inside. So they, because the head is bigger, it requires bigger space compared to the tail. So they will also carve the protein. So there are many examples known. I have just given one name, carved bar proteins, pretty common in biology. And then there are possibilities that a membrane can be deformed by a bundle of these actin filaments pushing from inside. Okay, it's just like a rod. You are pushing a rod. And you have put, let's say, you have put a stick inside a balloon, and because of the stick, the balloon will try to project out. So something similar to that. So I am going to show you a very particular experiment where you will see that tubes coming out from vesicles. So a spherical object, the vesicle, initially, and because of certain protein sitting on its surface, it will give out tubes. Okay, so here is the experiment. So this is the vesicle that you are seeing. The bright thing is the surface of it, fluorescently lit. And because of certain proteins, which you don't see, which are put on the outer surface, the vesicle gives out these tubes. Okay, these projected things are the tubes. Sorry. Okay, and these tubes are narrow, like nanometers, 50 nanometers or so, whereas the vesicles are quite big. I mean, these are real diagrams, and this is in minutes. So with time, the tubes grow, and if you stop supplying GTP, the tubes shrink. This is a very particular protein called FTSZ, which is relevant for bacteria, which you don't have to care. Some protein put on a vesicle giving rise to tubes. Okay, that's the summary. Now, question is, of course, how do you get these tubes? Okay. Now, typically, the examples that I gave you earlier, these kind of examples are common, that there are certain proteins which are carved, which are not straight, which like to basically they like to be on a curved membrane, and because of that, their own curvature, they are forcing the membrane to be curved. Hence, this tube is coming out on whose surface the curved protein will be happy because it's curved. Okay, so it's sort of strangling the membrane and hence the tube. But in this particular experiment, these proteins, curved proteins, don't arrange like this. They arrange straight, which is a surprise. Okay, so that required a new model to explain that. So that brings me to membrane models. Okay. Yeah. Yeah.
This kind of, rather than pushing, it's actually the other way around.
Yes. So this is from outside. So instead of some bundle of filaments pushing from inside, this is from outside. So these proteins are put on the outside. Okay, now I come to a little bit of membrane physics. Now, one thing I should tell you is that I'm showing lots of pictures and I am really waving my hand. I'm not coming to the mathematics purposefully. But underlying all this, there is a very vivid mathematical description to handle this membrane, polymers, and all these. Okay, which one has to know, but cannot talk about in detail often because it's cumbersome. Okay, so this is just a second. Yeah, so I'm coming to a bit of membrane physics after this. So some more observation here. If you look closely, the cross section of these vesicles, you see this kind of structure. So you see these dips here, and you don't see the tubes which come out from the junctions actually, where these two concave deep meets. Okay, and this is from the upper surface of the vesicle. These lighted objects are the tubes. And if you look at the arrangement of these tubes or these bright spots, you see that this spot is surrounded by 5 others, so like a pentagon. Sorry, this one is surrounded by roughly 6. Then you will find another one which is surrounded by 5 and so on. So this surface has lots of pentagons and hexagons. This is a fluorescent microscopic picture. Fluorescent microscope, yeah. Okay. The whole thing is about 20 micron diameter. Okay, so this surface has a pattern. The tubes are not really random. And this has connection with soccer ball structure, which most of you from Bengal will know. I'm coming to that. Before that, how do I describe the membranes? I said that membranes is a double layer. Okay. Now, a membrane which is like this is from this book, quite a famous biophysics book, very popular. So they can deform in various ways. They can stretch, they can bend, the thickness can change, they may get pressed, or they may glide, shear. Okay. Now the kind of membranes I am talking about, including the ones which are on the cell, their stretching elasticity or this compression elasticity is very, very high. You can hardly deform them. Okay. On the other hand, their shear elasticity is 0 because, and that's why they are called fluid membrane, in the sense that these molecules which make the membrane, they can diffuse freely, and these two layers also very easily glide past each other. That's why they are called fluid membrane. Okay. So unlike elastic layers, there is no shear modulus here. Whereas their bending rigidity is rather low. You can very easily bend them, and hence it is easy to change their shape. Okay. So these are all very engineering kind of material. I mean, this kind of picture you don't find in physics books, other than Rob Phillips' book. And the underlying model by which you model a membrane is the following. So this is a sheet, let's say an elastic sheet, a surface, okay, curved surface, very popular with gravitation people, for example, and many other type of physicists to mathematicians. So this is a curved surface. You want to describe the shape of the curved surface. So standard technology is, I mean, standard math is there for that. You have to define curvature matrix. So curvature matrix is nothing but the gradient of the normal vector. So at each point on this surface, there is a normal vector, and this curvature matrix gives you how the normal vector is changing in space. Okay, but it depends on which direction it is changing. Whether it's, let's say I have x in this direction, y in that direction. So that's why it's a matrix. I and j both can be x and y, or two variables on this surface. Okay, so it basically tells you this matrix has the information about how this normal is changing. Okay, so this matrix has two nice eigenvalues, and these eigenvalues actually gives you the curvature along two directions. Now you can imagine that through a point, you can pass many circles, infinite number of circles through that point. Okay, these are 3D curved surface. But among all those, the maximum radius and the minimum radius are the inverse of C1 and C2. Okay, so there is a theorem which tells you that the two eigenvalues of this curvature matrix are the two principal curvatures. That means the direction along which at a given point the curvature is the maximum, and orthogonal to that will be, okay, not always orthogonal, but the other one corresponds to the other direction through which it is minimal. So for example, think of a cylinder. At a given point on the cylinder, on one direction it's straight, so it is radius of curvature is infinity, so curvature is 0. Other direction, the curved direction, it is some finite. Okay, so for a cylinder, these are the two principal curvatures. For a sphere, any direction you go, it's the same, which is the 1 by r of the sphere. So thing is that when you have to write down the bending energy of this membrane, this has to be in terms of invariance of this curvature matrix, and sometimes maybe this normal vector. Why is that? Because Hamiltonian is basically energy. It should not depend on frame, how you put your XYZ frame. Okay, so it should be frame independent. So a Hamiltonian should be invariant with respect to any frame change, any rotation of frames. That's why you can only put in traces and determinants of this curvature matrix into the Hamiltonian. Okay, and you see there are squares here because see this trace is addition of these two curvatures, but because it's square, whether you bend the membrane this direction or that direction doesn't matter. They have the same energy. But sometimes membranes don't want to be flat, meaning their lowest energy is not flat. They want to be curved, let's say. In that case, you have to get in intrinsic curvature, and that breaks the up-down symmetry, meaning convex is not same as convex. They have different energies. Okay, so this is intrinsic curvature. But still you can show that this system, see this is after all a constant, so still the Hamiltonian is function of the traces only, which is independent of any particular coordinate frame. Okay, but because you are breaking the symmetry, certain things will happen. Okay, so that's a very brief description of membranes. Okay, so I already explained to you by what these circles means, the curvature in different directions. Now I told you that I am putting in proteins on this membrane, which brings in the change in the shape. Okay, so these are the cartoons for the proteins. So they are rod-like molecules called nematic liquid crystals. You see the head, there is no head and tail. That's why nematic. And these rod-like molecules put in bulk, they can form various phases depending on temperature and other effects. So they can form an isotropic liquid type state, or they can form a liquid crystalline state. By liquid crystalline, I mean, if you look at the rods, they are preferentially oriented along one direction. Although they are, if you look at their center of masses, it's quite random, but they are preferentially oriented along this direction. This is a crystalline solid, it's called a smectic state, again made of liquid crystals, but there you see a sort of a order. I mean, not only orientation, but they are all in planes. So the mass density, if you look at the mass density, there will be periodicity in this direction. Okay, so we will be mainly concerned with this particular phase. Okay, so now I come to the modeling of the particular experiment which I told you. That why does the membrane or the vesicle gives out tubes? Okay, so in order to model this, what we do is we cover the surface with a grid, okay, triangular grid, and at every grid point, at every vertex point, we put a rod-like protein, which has a curvature which you don't see because it's very, very small compared to the size of the membrane, size of the vesicle. But what they do is they try to impose some curvature on the vesicle at that point. Okay. Now, these two figures are not really one-to-one because, I mean, I have so many vertices here, but there are much less number of protein. I mean, they're not really one-to-one. It is just a cartoon that how we construct the mesh. Here also, there is an underlying mesh which you don't see, but this mesh, the triangles are much bigger, and at every vertex you have one such rod. Okay. So now when the proteins don't have any interactions between them, the rods, then they will not do anything to the vesicle. They will just sit onto it happily. But now I said that these proteins are not that innocent. They are curved and they want to impose their curvature on the membrane. Okay, so for that something will happen. So these proteins are given by their orientation of their axis, and then I have the curvature field by which I define the membrane free energy, membrane energy, and it's a closed membrane. That's also important. Now I am going to show you a model which was actually proposed by Sunil Kumar, who is at IIT Madras, and his collaborator Ramkrishnan was a PhD student that time. So we are using this grid, the way they formed the grid and their technology to explain our system. Okay, we are just applying this methodology to our system. Okay, so this is the new Hamiltonian. So what it is doing is that it is saying that earlier you saw that it was just C1 plus C2 whole square, that is curvature square. Okay, two principal curvature square. Here what I am saying is I should explain what is what. So think of this just one particular point at the vertex. So I have a whole closed surface. This is just one particular vertex, and at that vertex a protein is sitting, this blue rod. Now this it can sit along any direction, but it is sitting in the tangent plane, this ash surface that you see, small one. So what I'm saying is that we know that these proteins sit on the surface. Okay, so it is in the discrete language they're sitting on the tangent plane. Now at every point there is a curvature matrix. It has two eigenvectors and two eigenvalues and two eigenvectors. The two eigenvectors are these two red vectors that you see. Okay, so this protein, so it may not sit along this T1 and T2. It may sit somewhere in between. So physically, what I expect is that along its length, the protein, it will tend to curve the membrane. Okay, so if this is the rod sitting on the membrane which is there, it will tend to bend the rod in this direction along its length. Okay, not perpendicular to its length. So that information has gone in here, that this is the local curvature along the length of the rod, and this is the intrinsic curvature. That is what the rod wants to impose on the membrane. Okay, so in order to minimize this term, the curvature along the rod should match with the curvature of the protein. I mean, curvature of the membrane should match with the curvature of the protein in order to minimize this. Okay, and there is another term which is perpendicular to the rod. I also have an intrinsic curvature. This is somewhat ad hoc, but we will see that, I mean, physically it doesn't make sense at this point, but we will see that the effect that I'm going to tell you is all because of this effect, and I'll give you the reason. And also these proteins that are sitting on the surface, they are interacting with each other with nematic interaction. So rod-rod interaction, the tendency is to remain parallel. Okay, so I am in the nematic regime. I am not in the isotropic regime. Okay, so with this and with a non-zero value of this C perpendicular, you will see what will happen. Okay, by the way, I should tell you that we are putting in terms by hand, but all this has to obey the basic principle that your Hamiltonian should be invariant with respect to traces and determinants. Meaning, it should be invariant with respect to any rotation. Hence, it should be function of traces and determinants of this curvature matrix and maybe these vectors. So one can show this H parallel and H perpendicular. They can be written like traces with complicated matrix, curvature matrix, director, vector, and so on. But they are traces. So one can show that they are traces. And hence, we are not doing something which is not allowed by rotational invariance. Okay. So this is a simulation. We are starting with a roundish object. It is not really, it looks like a sphere, but actually the area is little more than a sphere. Okay, it is little soft.
Is that dependent on the triangular mesh or is it general?
Okay, triangular mesh is just a grid, meaning it is whenever you have a closed surface, triangular meshes are easy to close. If you take a square mesh, it will be more difficult. So it is, the physics is not dependent on the mesh, and also the size of the mesh and all that. So we have to make sure that it should not depend on those things. Yes.
So the Hamiltonian of that structure.
Yes. Yes. The Hamiltonian is actually a continuous Hamiltonian, but we discretize that on a mesh. Okay. So let me see. Yeah. See, this is what happens. I will explain to you why that is happening. Have I crossed half time? Time wise? Or 50? Is it? When did I start? 50 something. No, I don't remember. Anyway. Okay. Okay, so you see that tubes are occurring, and the orientation is along the length, as you saw in the experiment. Not everywhere, on the tubes. Okay, and in between, so you see these valleys and so on. Okay, so in nutshell, what is happening is this: you start from this vesicle. Depending on these parameters, these intrinsic curvatures that you have put in, it can go to this shape, it can go to this shape also, that is tube growing inward, and this has been seen. I didn't show you the diagram just to not overload you. So both type of tubes have been seen depending on how you attach these proteins on the membrane. Okay, so I come directly to this particular C perpendicular, which I was saying that it is ad hoc. So what this term is doing, it is actually helping this tube formation. Why? Is that, just think of this structure. Suppose if the rods align like this around the tube, then you can see that there is a curvature in the perpendicular direction. Okay, and by choosing this C perpendicular to be large, that is high curved, you can generate these tubes. So these tubes are, in a way, you can interpret them as bundling of these filaments. You see that in a very short radius, all these filaments are straight. Okay. Now in this model, whether these rods are inside the tube or outside the tube doesn't matter. Sorry, yeah, outside the membrane or inside the membrane doesn't matter. So this tantamounts to, this is a mimicry for bundle formation. Okay, so which didn't make any sense this term at the beginning, but this is actually a proxy for bundle formation. So I am promoting bundle formation everywhere, and hence these tubes form. And these tubes actually do not arrange randomly. And the reason is that if you look at on this vesicle, this very well-defined structure, you will see that near the tube, they are longitudinal, and near the tip of the tube, they're like star-shaped, meaning from the top point, they're going in all directions. So these are actually defects. Here also, you see certain structures, these three rods together, that is also a defect. Now, in the liquid crystalline language, on a 2D surface, if you try to arrange rods, one can form the various type of defects, these black spots. Okay, and there is a way to define their charge, topological charge. Okay, this is not real charge. That is, if you go around the loop, how much does the rod rotate? That sort of thing. And according to that, you can attach these different kind of charges to these defects. And there is a theorem due to Poincare long time back. It says that the total charge of these defects on any surface of genus G is 2 minus G. So for example, a sphere has genus 0. So genus is basically how many handles do you have or how many holes do you have. So a closed surface doesn't have any hole. On the other hand, this cup that you see, this coffee cup has one handle. So that has a genus 2. So on such a surface with genus 0, the total charge should be 2. Okay, so this is a constraint coming from this theorem. And then when you go back and try to look at these shapes, what you find is that it exactly satisfies that theorem. There is no escape from a theorem after all. So Now I'll just try to explain. So of course I didn't know about all these things. After we saw these structures, we started scratching our head, and then we found such things people knew long time. Okay, so this point is a +1 defect. So this whole surface, you can just put pentagons and hexagon and cover this surface. For example, this peak around it has 5 valleys. Each valley has a -1/2 defect, and the peak has a +1 defect. So this is a pentagon. So if you count that how much defect belongs to this pentagon, then what you have to do is that this plus defect is fully belonging to this pentagon, whereas this -1/2 defect belong to 3 other tubes. Okay, one here and on the 2 sides. So these -1/2 defects belong to 3 other tubes, so hence 1/3, and there are 5 of them. The total is 1/6. Whereas if this was a hexagon, this number will be 0. Now it turns out that a closed surface like this, let's say a soccer ball spherical structure, can be covered not by hexagons only. Minimum 12 pentagons and 20 hexagons. That is the minimal structure. So any soccer ball you pick up, which old time ones which used to have these pentagons, hexagons, you will see these structures, total 32 squares. You can have, 32 patches. You can have more actually, but there also this number is somewhat quantized. Any number is not possible. It's coming from the topology of the surface. Okay. So if you use that information, 12 pentagons each giving 1/6 gives you exactly 2. Hexagons doesn't give anything. Okay. So this structure is not a random structure. The tubes maintain sort of equal distance from each other, and it obeys the topology. Okay. Now, so this is an example, a classical example, where a theorem which tells you about the global amount of charge that the system should have, topological charge, not real charge. It dictates, it controls what the local structure will be, what will be the let's say inter-tube distance or inter-vertex distance, wherever you like. There's a very loose analogy. You might have heard these days about topological material, topological quantum material. So their topology in those cases protect quantum states, meaning, and they are very useful in quantum. People are exploring them for quantum computers, because topological constraint would like to preserve the quantum state, doesn't let it decay. And decorrelation is a major problem in quantum computation. Okay, so people are exploring how this quantum material using topological states, how can you generate quantum? How can you have quantum computing and so on? This is just a so one similar question here will be to understand that. Of course, you can see that this structure is being maintained by this theorem. So you can engineer around this theorem and develop structures which will still have a controlled number of hexagons and pentagons. This is just a wild simile. Okay, so I've already told you. Now here is another application. I will not go in the details of this these days, because activity and active physics is very popular. For Hydra, earlier people had been studying Hydra for developmental biology point of view. They're very popular because if you cut it, so this is a Hydra. All of you know what a Hydra is in biology books, right? They're known for somersaulting and so on. The great thing about Hydra is that if you cut a piece from anywhere and let it live, just let it in water, it will grow the whole Hydra from there, almost like stem cells. Okay, so developmental biologists have been very interested how that happens. Organ replacement and all that will be greatly helped if you understood that. Now, physicists recently looked at Hydra using their fluorescent microscopes, and they found that these filaments, actin filaments beneath their body, is very well arranged. Okay, you can see they're all aligned, and they showed that this also has these defects. One at the top, which they call the mouth. So these are very rigid, the mouth and foot portion, +1s, and each tentacle are charge neutral. That is a +1 defect here and 2 -1 defects here. Okay. So even without the tentacles also, it has total +2 according to that theorem, because it's a closed surface. Okay. Although one big difference here is that it is not really small filaments like pneumatic rods. It is rather long growing actin filaments. Okay, so it's the physics may not be exactly similar to liquid crystal physics if you try that. Okay, so we tried just our poor man's way of modeling, trying to answer the question: how important is it to have this mouth here, that is +1 defect for the tentacles? Because this paper advertised about that this placement of this mouth is very important. So all that we did was we took a closed vesicle and we so this is the cylindrical portion. So we didn't allow anything there. Very strong, elastically rigid, strong bending rigidity in this portion. Upper portion we allowed flexibility, and it is known that there is, in case of Hydra, there is a particular chemical called beta-catenin, which physicists don't care much about, but biologists know that that's all important. That is the reason for giving rise to this bundle, these tentacles, because this protein actually promote bundling of actin. Okay, and these actins grow and make these tentacles. So we said that, okay, in this region, the upper portion, the head, we maintained the +1 defect here by hand. So in a very small area, we maintained the +1 defect by holding the arrangement of the rods fixed there, and we let whatever it wants to do in the neighborhood, and we applied C perpendicular term, which gave us tubes. Okay, so not very unexpected. You see the tubes here, but the main goal was to see that is having this +1 defect. Yeah,
Complete within 3 or 4 minutes.
complete 3 and 4 minutes, really? Okay. Oh, okay, fine. So what we found was that no, that particular +1 defect is not really crucial. You can have other type of defects also. This is also +1, but different configuration. But you can still have these tubes. Okay, so having the defect is not really very important here. It is just a consequence that you cannot arrange the. Okay, so there are other applications of this we have done, but I'll skip that. So summary 1, this was supposed to be the half, but anyway. Okay, so the main conclusion is the defect is not the driver, but it's a consequence of the surface topology. I don't know. It is Hydra matures in days actually, not in hours. But mature meaning that this head region becomes very rigid. Maturity means that. Okay, so this is the second part I wanted to talk about, but okay, there is not much time. I'll take 3-4 minutes only. So Abhishek told you a lot about active matter. Okay, I mean that these filaments which are below the cell surface, I said they flow, and they are described by active matter, namely this Q alpha beta tensor. They capture the alignment. So when the cell divides, a wall forms in the middle, and you can describe the motion of these elements in that wall. That means when the wall forms, it forms like this, like the shutter of a camera. So the flow of material here, so you can abstract this as a 2D. If you just look at the 2D cross section of this, it will look like this with a hole inside, and the walls are caving in from the sides. So here is the actin, which is like a gel which is flowing inward. So this physics you can describe using active matter, and because it's a polar geometry, 2D, highly simplified, with relevant boundary condition, you can actually solve exactly. And so these are the people who actually worked on this. Amitabh is a colleague. He was a PhD student. He is now in Kolkata, and he is still doing PhD. He was our undergrad student. So I'll just, okay, so I don't have time to explain to you how does activity actually generate flows. I said activity generates stresses, okay, and the stress drives flow. I will directly take you to a simulation. So this was the exact calculation for that 2D polar thing. So you can do all that, and this is already there, published. But now we wanted to actually look at the flows on the surface, not at the cross section. So for that, we had set up, using this active matter theory, a simulation of these equations. These are called phase field equation. There is a order velocity field, there is a order parameter field, u is the velocity, qij is the order parameter, that means the orientation of these filaments, and phi is the phase, which tells you that whether you are inside the cell or outside the cell. So it's pretty, it is the same set of equation which Abhishek was showing you. I will not go into detail. All that we do here is that we, by this simulation, we avoid dealing with the interface and we deal it in an indirect way. Okay, so that's one simplification. I mean, one usefulness of this kind of simulation. I'll directly show you what kind of motion you see. And when the cell division occurs, before that, what happens is that flows come from the two polar regions towards the middle. And it is because in the middle, the activity is very high. So that pulls flow from two sides. Okay, so this flow will be happening on a curved surface. So in nutshell, this is the, I will show you one simulation. Maybe that will show you the flow. Okay, this is the orientation of these rods. Sorry, this is the velocity field that you are seeing. The flow is happening on a curved surface. And you see that slowly notch is occurring at the two ends. I'll shortcut this. Here, all the velocity vectors, irrespective of their magnitude, have been given the, I mean, I am putting a velocity vector of unit magnitude for all of them, irrespective of their magnitude. But actually, in reality, flows are very strong here, very weak near the boundaries. There is this picture will tell you that story, that flows are actually very strong in the middle and very small at the two boundaries. And so this is the flow, and next is the, how does the nematic rods orient? And this is something interesting, that they are aligned parallel direction, but at the middle, they align in the perpendicular direction. So it's forming a ring actually. Okay, and this ring is responsible for constricting it in the middle. So we are trying to understand the physics of this ring, that why this is forming. Flows are occurring from the two sides towards the middle, and because of that, the surface is going inward. So we are trying to understand it a little more quantitatively, just by this simple model, that if you have two mediums, one medium below, one medium above, just like the cell, inside and inside, inside and outside, and flow is coming towards the middle from the two sides. So it's a compressive flow. So because fluid mechanics has to obey continuity, where will the flow go? It cannot vanish. So either it can come to zero or it can go in. Okay. So what you see here is that's what is happening, even for flat surfaces. So this is a buckling phenomena. You can see here, you don't see the flow here. You just see the rods here. They are aligned here along the parallel direction, and that will mean a ring formation at the middle. So this buckling doesn't have to do anything with the curvature of the surface. Okay, so that's one thing we have understood, but we are trying to set up a exact calculation to understand this buckling. Okay, so that's it. This is the summary. So we are understanding part of that now. How does this buckling takes place? I didn't tell you much about this ring, because this ring is very important. I showed you, I think, some pictures at the beginning. And the flow has lots of details, which I don't want to go into. Yeah, so this ring, I just wanted to show you this. Yeah, this ring is very important. It will figure in any cell division context. Because of this ring, that means a concentrated bundle of actin, which forms at the middle and it pulls. Okay, just like a rubber band. Okay, so I'll just stop here. Thank you.
So excellent talk, and there is a lot of possibility of working on this field. Now we allow only 4 questions from the audience.
Any question?
Sir, I have a question.
Yeah.
That you have talked about pneumatic phase, order phase and disordered phase.
Yes.
So can we get the phase transition when it goes?
No. See, here we are working completely in the nematic phase, because actins are known to align mostly. near the cell division site. So we are assuming we are already in that regime, and what happens to that because of the flows. Okay. Sorry, I should correct myself slightly. Sorry. No, sorry. Rather, I am always in the isotropic phase. Because of the flow, it gets aligned. Okay, so that's why away from it, it always remains unaligned, but at the middle, because of the flow, it gets aligned and form the ring, etc. Yeah.
Any other?
What is this contractile force? See, I mean, just think of a rubber band. If you expand the rubber band, then what does it want to do? It wants to shrink, right? My hands inward, right?
And balloon also. The balloon is
Yes, true. Correct. Now the thing is that who makes the rubber band for the cell? So these actins are bundling up and making that rubber band and pulling. But there's little more to that. When the rubber band shrinks, it does not lose material. Here, this ring is not one cable. It is many small rod-type proteins. They are sort of connecting up because of their actin, myosin, and linker, etc., and they are constantly shedding material and gathering material by polymerization. So it's a dynamic thing which is shrinking like a rubber band, but it's not really a rubber band. Yeah. Abhishek had a question here. You can just say.
So, in the first part of your talk, there is maybe a distract, but these rods on the, when you're looking at the center of mass, right, and when you're saying that it is acting on the vertex, it could be as well like the orientation, right? I mean, along the orientation of this.
The rod is in the tangent plane of the vertex.
It is in the
tangent plane.
Because it cannot be on the, it cannot lie on the sphere.
No.
Right? So,
and also it is imposing a curvature on the membrane. But in reality, it itself is also getting curved. But we are not. See, we want to see what it does to the membrane. Okay.
It's not possible to include the curvature of.
Yeah. So, but I have included their effect that they want to curve the membrane. That effect is there. I can take the rods and draw curved proteins there. That's just a visualization.
And the excluded volume interaction is kind of automatically there. Right?
Excluded volume.
For the vortex model.
So this is not a vertex model. This is.
I'm saying that
It is on the surface. You have a protein, so we don't take care of.
the fact that
It is sitting on it, so nobody is obstructing each other. Yeah.
Because even inside the cell, that would also matter, right? I mean, the fact that they can.
Inside the cell.
Yeah, I mean, on the when what you're trying to say is that just below the cell surface.
That actin, yeah.
Actin cortex.
Yes.
Their
excluded volume will matter. Depends on the density. Depends on. Of course, this actin is known to be a very dirty network of cross-linking things.
Yeah.
We just model that in a simple way using this nematic language. It captures the physics, but it is not real. But it captures the physics.
Okay.
Any other question?
Yeah.
Sir, at first you showed the Hamiltonian with trace of CIT and development of CIT only.
Yeah.
What sort of shape does that Hamiltonian prefer?
What sort of?
What sort of shape does it prefer?
Shape.
Yes.
Okay, that depends. Okay, if you don't have any C0, meaning any intrinsic curvature, then the lowest energy state is the flat state. Because if you see, sorry, where is that? If you see the Hamiltonian, see the first term, trace square, positive definite. So minimum value can be 0. So you can have C1 plus C2 equal to 0. Okay. You can. So one simple possibility is C1, C2 both are 0. Meaning that means curvature is 0, that means flat. That will minimize this term also. But okay, for closed vesicle, this term actually doesn't matter for a different reason. So this will prefer a flat state. You can also have C1 plus C2 equal to 0, but individually they are not 0, plus and minus. That state is called a catenoid state. It's a very special state known, but yeah, so that also have zero energy given this Hamiltonian, but you don't see that in real membrane that requires certain kind of connectivity and so on. But one line answer is flat state. Sorry?
The determinant term.
Okay, the determinant. The determinant is, okay, the coefficient of this is called a Gaussian curvature. Okay. So if you have a closed surface, then it turns out that if you do an area integral of this term, it's a constant. Gauss-Bonnet theorem. But for a flat surface, see, I said that its bending effect is completely captured by this. That's not true. Bending effect is there in both. For example, suppose I have an open surface, C1 and C2, their sum is 0. So this sum is 0. Okay. But if these C1 and C2 have very high values, then this term will come in, right? It can become positive, negative, whatever. You don't want that. So, but the point is that this also has an effect.
So at the end, I have got very small.
Please.
So what is the actually driving force for the cell division? So mainly you have shown some mechanical force.
I showed only the
factor.
I showed only the actually what I showed is the very last stage of cell division where the cell is physically dividing between two parts. But before that, everything is biochemistry. Like there was a nucleus which divided into two, spindle etc. formed. Spindle went to the two sides. All biochemistry, chemical signaling.
Now, what are the supposed effect of surface tension or any other electrostatic force? This and that. Are they playing any role? Vital
Electrostatic,
role?
I don't think so. Surface tension plays a role in keeping the shape of the cell intact because because of this network of proteins beneath that there is a surface tension, and that tends to keep it prevents the surface from bloating up. So what that does is it's like an elastic sheet, and it is trying to reduce its area because of the surface tension, and inside you have a pressure. So they are countering each other. So if you make a hole because of this pressure, the liquid will just leak out. So surface tension has a role. Yeah.
Okay, so if there is no any question,
Oh, just a second. Up down symmetry. Where is it? Yeah.
And back side also. Up down symmetry
Yeah. See.
at the very end.
Yes. Okay. It is very simple. Suppose it is saying there is a difference between the energy of a convex and concave surface will be different if this term is there. Okay. If this term is not there, okay. So let's say think of a sphere, convex, C1 and C2, both curvatures are positive. Okay. So because I have this square, all that matters is C1 plus C2 square. If that same surface instead of convex, if it is concave, then C1 and C2 both are negative. But because of this square, again it is coming to C1 plus. So convex and concave have same energy if this intrinsic curvature term is not there. Okay. But if the sphere wants to be a convex, meaning the surface wants to be convex because of some underlying other reasons, then these two are not equivalent. They do not have same energy. So in that sense, up-down symmetry.
okay.
Yeah.
So thank you very much
Thank
for
you.
for an exciting talk. Now our undergraduate student or it will
Thank you, Shadrik. Welcome to our second oral presentation session. In this session, we have 5 speakers. A general reminder to all, this session will essentially be a 12-minute of talk and a 3-minute of question answer session. I will be reminding every presenter after 10 minutes. Thank you. Now, I would like to invite on stage the first speaker, Dr. Shrabanti Dhar. She is a state-aided college teacher at Gokhale Memorial Girls College, Kolkata. Over to you. Good afternoon, everyone. Well, I would like to thank the organizers for allowing me to present my work. So, the title of my work is First Passage Phenomena in Quantum Systems. And this work has been done in collaboration with Professor Shrabanti. He is presently at HRI, Allahabad, Professor Abhishek Dhar, and Professor Diptiman Singh. So this is the flowchart of my talk. So first I will introduce the classical first passage problem and then the framework of the uh, quantum problem, and then I will explain the projective measurements, where I will explain the protocol of our measurement scheme, followed by the results and summary. Okay, so first passage problem, it basically deals with the probability with which a stochastic or a random process, it first encounters a threshold. So if you start with a very simple 1D random walker, so here we have a walker which how, how does it?
Originally, you can take from here.
So here we have a random walker which starts from the origin and it goes to left or right with equal probability. And so if we ask that the probability that this walker will reach at the site X after T steps, it is called the occupation probability and it varies as 1 upon root T. Now this occupation probability, it actually includes all the first, second and third visits to this point X. Now, what is this first passage probability? So if you ask the question that the probability that this walker will reach this point X after T steps for the first time only, so then this probability, it varies as 1 by T to the power 3 by 2. Now, this first passage phenomena is also studied in the context of chemistry, biology, and also in some socio-economic problems. So, what is this quantum first passage problem? So, here we ask the question that when will a quantum particle which is released from a fixed point in space, it will be detected for the first time at some point in its vicinity. So it is basically a time of arrival problem, and we are actually measuring the time in a quantum system, which is quite a difficult problem because of the reasons that there are some controversies associated with defining a Hermitian operator associated with time, and also from the measurement postulate of quantum mechanics, we know that any sort of measurement on a quantum system, it actually alters the state of the system. So it's a bit difficult problem to handle. So here we have considered a lattice version of a free particle. So this particle is initially prepared in some initial state, okay, and it is allowed to hop on this lattice following this nearest neighbor tight binding Hamiltonian. And we ask that, and here at the nth site we have kept a detector, and this detector, it keeps on making instantaneous detections at regular finite intervals of time until it detects this particle for the first time, and we have called this time as the first detection time. Okay. Now, this time t is basically a stochastic variable, and so here we have actually determined the complementary probability of non-detection during each measurement. That is, we have basically studied the survival probability of the particle, that it will be detected only at this time t, and it will not be detected at any time before this time t. So this survival probability, it represents the probability that this particle, it remains undetected till the time t. Now, to measure this survival probability, we have defined two projection operators. Now, we can, in a very simple way, we can say that projection is like extracting out some component of this wave function psi. So we can, in a very simple way, understand this concept of projection. So if we define a matrix like this, and if we operate this matrix on a 3D vector, so we see that this matrix, it projects the vector on a 2D plane. Its third component, it becomes 0. So for our work, we have defined two projection operators. One is A, which corresponds to detecting the particle at the site n. So this A is represented as n. It is basically an outer product of n. So this n, it basically represents a column vector with 1 at the nth position of this vector, and it is 0 elsewhere. That means the particle will be localized at that point only. So now, after the detection, the probability of the detection, it will be given by n psi naught squared, which is basically the expectation value of this projection operator in the state psi. And after the measurement, if the particle is detected, the state collapses to this, the system collapses to this state. And we have defined a complementary operator, which is b equal to 1 minus a. So this operator, it actually makes the nth element of psi 0, as I had showed in the previous slide. And probability of non-detection, or the survival probability, which we are trying to measure here, will be given by the expectation value of this complementary operator in the state psi, which is equal to 1 minus the detection probability. So if the measurement doesn't detect the particle, then the wave function, it immediately projects to the state psi plus. Now this plus means the wave function after the measurement, in case the particle is not detected. So here we just explain the protocol of our experiment. we have considered a replica of our experiment. So all these replica at time t equal to 0, all these dots are actually blue. Okay, so we allow each system to evolve with time following this Hamiltonian, and then we make an instantaneous measurement at time t equal to tau. Now, suppose that in these green dots, the particle has been detected, and in the blue dots, it is still undetected. Again, we allow the system to evolve with time, and then we observe that in these red dots, the particle is detected, and in the blue ones, it is still undetected. Okay, so in the step 1, we allow the particle to evolve with time in each realization, and then in step 2, we carry out the detection or the elimination process. That what we will do is, in the ensemble or in the members of the ensemble where this particle will be detected, we will just discard those realizations. Okay, so the state just after the measurement where this particle will not be detected is given by psi1 plus, which is basically equal to this B operator operating on the wave function after the evolution. Okay. So this is a very simple a pictorial representation. So we start with this particle at time t equal to 0, where it is allowed to evolve with time, and these are the possible states after measurement if the particle is not detected and if the particle is detected. So the steps 1 and 2, that is the yes, and that is the evolution and the elimination. It can be represented mathematically in this way. So it is the wave function just before the nth measurement, and this is the wave function just after the measurement, just after the nth measurement. So the survival probability after the nth measurement will be given by this Pn, which is the inner product of the wave function after the measurement. And this, the first detection probability is the difference of these survival probabilities in the n minus 1th and the nth measurement. So we can represent a very generalized Hamiltonian in this way, where this Hs represents the Hamiltonian of the system, Hm the Hamiltonian of the measuring device, and this V represents the coupling between the system and the measuring device. Okay. So here, to find the survival probability, we have to find this psi n plus, and in order to find psi n plus, we have to diagonalize this quantity u tilde, which is bub. Now, this exact diagonalization of this operator is very difficult. So here we have used perturbation theory. We assume that this time interval is very small. That is, the time interval between successive measurements is very small as compared to the change of the wave function, and so we retain up to the quadratic term in tau, and we observe that the evolution of the system, it is actually effectively controlled by a non-Hermitian Hamiltonian. So here H effective is equal to Hs plus V effective. So this represents the evolution by a non-Hermitian Hamiltonian. So for this 1D case, our system Hamiltonian is given by this expression, and the effective.
Please conclude.
So, and this effective potential is this, and So here we have discussed two initial conditions, that when the initial state is an eigenstate of our system Hamiltonian, we have obtained an exponential decay. And when it is a position eigenstate, we have obtained a power law decay of the survival probability. So this is our result. We have obtained a power law decay, and at times of the order of n cube, this converts to an exponential decay. And we have also investigated ring geometry, where we have obtained similar power law decay. So this is the summary. So here we have addressed a typical time of arrival problem, and we observe that the measurement alters the state of the system. So it is a non-trivial problem, where we have considered a situation where a device makes instantaneous measurements, and with this perturbation theory, we obtained a power law behavior at the asymptotic limits. And cold atoms on optical lattices, it can serve as ideal experiments where these quantum mechanical predictions can be tested. So that's all. Thank you.
Thank you, Dr. Shrabanti Dhar. Moving on to the question and answer session. Any questions?
Moving to the first motivation part, that you are just taking a random walker and just checking out the one-sidedness. That he is only moving to that side. That part of the probability you are just calculating. Can you just clarify why is that only one side is only calculated and the other side is probability is not?
That particle is, are you asking about the classical case?
Yes, but the very first, there are one is the 3 to the power half and the other one is the 3 to the power 3 by 2. So why is that 3 to the power 3
No,
by 2 is being
here, see, I have given the arrows to left and right both. So it can move either to left or to right with equal probability. And we have an absorber at this position X. Okay.
also surviving probability is for this absorber too. So after particle is detected, so that particle is now no more in the system.
Yes.
Oh,
Okay. Can we go to the last? Can we go to the last slide? Absolutely last one. The last point. Why do you think cold atoms on optical lattice could be a good experimental platform? And what is so special about cold atoms in optical lattice?
Means we actually thought that these systems can serve as ideal platform where the measurement postulates of these quantum mechanics, it can be verified.
Right. I'm just interested in knowing the reason behind your thinking. Why do you think they can be ideal systems? See, these are very specific words, cold atoms and optical
Yes.
lattices. I'm just curious. Anyway, we can just
So you consider only first nearest neighbor interaction.
Yes.
If you consider further nearest interaction, then the survival probability would be affected or your results. What would be that? Can you make any comment on that? If you consider further neighbor interaction, for example, say at this second neighbor,
Actually, we have considered a mean field model where this particle starting from any site, it can hop to any other set. So that we have investigated. So where there also we have obtained exponential decay of the survival.
is it the scale is same or scaling is same or like it?
In that case, in the mean field case, we have obtained exponential decay only, no power law decay.
Oh, exponential.
Yes.
And the second question is that does the survival probability depend on the site you are releasing the particle?
Yes.
So how do you then take care of that, because if suppose the wheel is in one side, then cyber probability say changes, then
Yeah, if it starts at the very boundary of the lattice, then it decays as 1 by t to the power half, and if it starts at the very bulk portion of the lattice, then it varies as 1 by t to the power of 3 by 2.
any side in the path or any specific.
Any site in the bulk or any site at the boundary.
Does that not mean that like it will also depend on the size of the system, like how many lattice points you have in your system? Because if you have a longer chain,
Yes.
then like the cyber probability will also decay.
Yes, since here tau, this time of interval is small enough, so yes, it will depend on the system size
So
also.
you have to accordingly you have to take
Yes.
like estimated size.
Yes.
You cannot go further beyond that. Then otherwise always cyber probability
Yes,
it will take a very long.
yes.
Just a follow-up question. Just a follow-up question is the so what is the first detection? What first detection that you have present?
First detection means it will be detected at the nth time only, and it will remain undetected up to time t equal to n minus 1 into tau. So it will be detected at n tau only, and it will remain undetected up to n minus 1 into tau. But tau is that finite time interval after which we are making the measurements. Time interval means so here so this is this time interval, this tau, and we have assumed here that this time interval is small as compared to the spread of the wave function. Okay, it is a finite time interval, but we have assumed that it is small as compared to the spread of the wave function over the lattice, and so we have used here the perturbative analysis to find a survival probability.
Thank you.
Thank you.
Allow me to invite our next speaker for this session, Ms. Ishita Tikader. She is a PhD scholar at the Department of Physics, Presidency University. Her topic of talk will be Monte Carlo study of magnetization in Ising ferromagnetic cubic shell. Over to you.
Thank you. Okay. Good afternoon, everyone. Today my talk on Monte Carlo study of magnetization in Ising ferromagnetic cubic shell. This is the outline of my talk I am going to discuss. And let's start with a simple but interesting question. What is the Euclidean dimension of cubic shell? Simple. We know that a cube is a 3D object, solid cube. Then just take out its inner bulk part, and it will make a hollow cube or cubic shell. Then what will be its dimension? If your answer is it is a 3D object, then can we expect that the magnetic properties in a Ising ferromagnetic cubic shell will show the behavior like a three-dimensional system? Let's think what happens. Here's the schematic description of cubic shell lattice. This is this blue one. Okay. These blue balls are the surface side on a cubic shell, and the red one defines the edge or boundary of the lattice. Okay, since we are going to discuss about the magnetic properties in Ising ferromagnetic cubic cell. So here is the standard Hamiltonian of Ising model, and G is the nearest neighbor interaction strength, and H is the external magnetic field. And since we are talking about Ising spin, so spin can take only one +1 or -1 value. We have conducted Monte Carlo-based simulation study with the Metropolis algorithm. According to Metropolis algorithm, the spin flip will be determined with probability. This is the minimum between 1 and exponential of this star, and this is the probability of the spin will flip or not. This is the spontaneous magnetization at each iteration or step. And this is the thermodynamic quantities in equilibrium system, the time average of magnetization and susceptibility. So, talking about magnetic properties, what is the magnetic property? Like behavior like phase transition or transient behavior such as relaxation behavior, metastability, and see what happens, the equilibrium or behavior of phase transition. Okay, study of phase transition. So we have here, so the variation of magnetization and susceptibility with temperature. And here's the comparative study between cubic cell and solid lattice. The peak of the susceptibility determines the critical, most can say the pseudo-critical temperature. What is the difference between pseudo-critical and critical? That is in a thermodynamic limit, but in terms to infinite, then we can say the critical point with phase transition occurs. This is the critical, and for finite system, size system, the susceptibility doesn't diverge. It just give a peak value. That's why we can define as the pseudo-critical. So for cubic cell, the pseudo-critical temperature gives us the 2.28 value, which is very much close to the value of Onsager value for TC for two-dimensional system. Onsager value for TC is 2.269, and we obtained the TC for cubic cell lattice of size equals to 100 is 2.28, where a solid cube gives us a pseudo-critical temperature 4.46, which is very much close to the computational value of 3D Ising system 4.511. And from this, it is clear that the phase transition in cubic cells behaves like a 2D system. It seems to be a surprising incident, but it is not, because even it is a 3D object, but we can see the lattice height, each lattice height, that each lattice size can interact with 4 nearest neighbor only. That means it's similar incident to the two-dimensional lattice. Okay. Now, from the equilibrium behavior, if we can see that it behaves like a two-dimensional system, then what happens the other behavior, magnetic properties like relaxation or metastable. Let's talk about the relaxation behavior. Okay. We have studied the decay of magnetization with time, both for cubic cell and solid cube, where it is clear that the solid cube exhibit the exponential relaxation from this. It is the linear plot, and this is the lower plot is the semi-log plot. If the relaxation is exponential nature, then its semi-log plot gives us a straight line, which is very much clear for the case for solid cube. But for cubic cell, it is not exponential behavior. But the theoretical studies, the previous theoretical and Monte Carlo simulation-based study, that those sites I can refer. They said that we can predict that we will get exponential relaxation for 2 and 3-dimensional Ising system near critical point. So this is quite interesting. It is neither behave like 2D, not 3D. For especially for relaxation behavior, we have also checked if it is stretched exponential or power law, but unfortunately not. If it is stretched exponential, then this figure gives us also a straight line, because log m versus t gives, if it gives a straight line, then we can conclude it is a stretched exponential, which is not. And also for the power law, it is not a power law relaxation. Now, let's see what happens in metastable behavior. What is metastability? Assume a ferromagnetic system, keep it in a ferro phase, that means lower than Tc, then all spins are aligned. That is state of magnetization 1. Now, apply a weak external field opposite to the direction of the orientation of the spin. Then what will happen? The system first try to keep itself in a metastable state of positive magnetization, and after a certain time, it will decay to the state of equilibrium with negative magnetization. And here is the, sorry, okay, here is the decay of metastable state. Before going to that, I will discuss about the classical nucleation theory of metastable state. This metastable behavior can be described by the classical nucleation theory using the growing and shrinking of the droplets. According to classical nucleation theory, the log of nucleation time varies as 1 over h x to the power 1 over h x to the power d minus 1, where d is the dimension of the system, and so that log gives us a linear relation, and the slope is different for 2 different regions. One region defined as a nucleation regime, and other one is defined as coalescence regime. Sorry, this is but the relation between the slope of coalescence regime and nucleation regime is this. The slope in nucleation regime is equal to d plus 1 times of the slope of coalescence regime. Now here is our result for decay of metastable state for cubic cell and solid cube. For cubic cell, multiple metastable state is observed, but there is only one single metastable state for cubic. That is, we can say that we can.
Sorry to interrupt you.
Okay, thank you. Okay, here is the distribution of nucleation time. Let me define what is the nucleation time. The minimum time iteration to achieve the state of below magnetization 0.7. We have shown for 2 different magnetic field, external field. For weaker magnetic field, the distribution of nucleation time is wider. That means there is an enormous fluctuation. We have checked the field dependence of the nucleation time. Tau nucleation is plotted against 1 by h and also 1 by h square in semi-log scale, and in both graphical plot, 3 different regime with different slope value is prominent here. One stronger field regime, this lower high field or stronger field regime. This is the intermediate field regime or coalescence regime, and this upper regime or weaker field regime or nucleation regime. Now, what will be its more? We are going to verify if it behaves like a 2D system or 3D. We are plotted for 2D. Log of tau nucleation is plotted against 1 by h, and we have fitted the slope for coalescence and both coalescence and nucleation regime. That is, here is the parameters. We have estimated the ratio between the slopes. Theoretically, for 2D, theoretical value of R is 3, that is D plus 1, but we have estimated, our estimated value of R is for lattice size 50, 2.28 and lattice size 40, 1.7 with error analysis this. And we have also plotted same thing for three-dimensional system where log of tau nucleation is plotted against 1 by h square. And here is the statistical analysis. And our statistical analysis clearly shows that nucleation in cubic cell more appreciably obeys the two-dimensional system than 3D. Here is the I can show the how nucleation occurs in coalescence regime or inter. Okay, 1 minute just. Okay, I think. Okay. Here in coalescence regime, two droplets march along the boundary, and for single droplet regime, one droplet grow at a time and continuously evolve and almost maximum part of the phase. And that's how this yellow part is for down up spins and the black part for down spin cluster. Is my I can conclude that the cubic cell shows the ferromagnetic phase transition near pseudo-critical temperature, which is very much close to the TC for two-dimensional system, and relaxation behavior doesn't exhibit neither exponential or stage exponential, neither not power law. And multiple metastable states are found in the case of decay of magnetization in cubic cell. Three distinct regime is very much clearly identified, and it's more behave like a two-dimensional system. And here's the reference. I would acknowledge my supervisor, Professor Mukti Sachit. Thank you.
Thank you, Ms. Ishita. Is there any other questions?
I have a question on the on-cellular variable.
Hmm.
The on-cellular temperature has any unit or it is just a ratio?
It's the unit of J by Kb unit. Kb means Boltzmann's constant and J is the interaction strength. Okay.
I just want to know what is this nucleation region and coalescence regions mean?
Actually, nucleation regime, this is determined by the slope value. Nucleation regime, there is one supercritical droplet. Supercritical size droplet means its first cluster grow. If its size critical value less than critical value, it shrinks another way, and if value is upper than the critical size, then it's grow. In nucleation regime or single droplet regime, one cluster grow and ultimately evolves the system. But in coalescence regime, two droplet or multiple droplet grow and simultaneously at the same time and emerge with each other.
Droplets you mean, as you were saying about the lattice thing.
I mean it's cluster of negative spins.
So it's droplets, it's like the lattice size atoms you can consider or something.
Yes. The lattice point which becoming downspin, cluster of downspin among the cluster of upspins.
Okay, thank you.
Next, I would like to invite Suvechha Indu. She is currently a senior research fellow at Calcutta University under Dr. Raka Dasgupta. Thank you.
Thank you so much for this nice introduction, and thanks to the organizers for giving me this opportunity to present my work in front of such large audiences. Today I shall talk about investigating the origin of the non-uniform phases in a bosonic Rydberg system. Firstly, I will talk about Rydberg atom and its properties. Then uh, I will describe the system of optical lattice with Rydberg atoms with many atoms per side. Uh, we have studied dynamics of the system with different phases and checked the stability of the fixed points of the system and have calculated the correlation with semi-classical Monte Carlo simulation. We have created different phases by tuning the interatomic interaction in a lattice site. What is Rydberg atom? Rydberg atom is an atom with one or more excited electrons having high principal quantum number n. This is a schematic diagram of Rydberg atom. This is nucleus, these are inner electrons, and this is Rydberg electron with high principal quantum number n. One of the main feature of Rydberg atom is Rydberg blockade. When two Rydberg atoms are in the same excited state, then dipole-dipole interaction acts between them and it shifts the neighboring energy level with an amount. So it is difficult for the nearer atom to go to the higher energy state as the energy gap increases. So one Rydberg excited atom prevents the excitation of the atoms of the nearer side. This is called Rydberg blockade interaction. Now this is our system. This is lower energy state and this is higher energy state. Here there are many atoms per site. When all the atoms are in the lower energy state, we call it ground state. And when one atom goes to the higher energy state, we call it excited state. This is on-site interaction U tilde, which is present among the atoms in the ground state of the same site. This delta tilde is the detuning. This is the energy gap between ground state and excited state. This omega tilde is the Rabi coupling, which acts between ground state and excited state. This gamma is dissipation coefficient, and V tilde is the nearest neighbor Rydberg interaction term. This is our Hamiltonian. This is detuning. This is Rabi coupling acts between E and G. For on-site interaction, we have two terms. One for ground state. When all the atoms are in the lower energy state, we take n into n minus 1. And when one atom goes to the higher energy state, we take n minus 1 into n minus 2. And this is Rydberg term. Here we sum over J. J is the site index and n is the number of bosons present in one site. For Rydberg term, we take sum over Jk. Jk is the nearest neighbor site index. From this Hamiltonian, we can get these dynamical equations rescaled by gamma, dissipation coefficient. Here, omega is the population imbalances between ground state and excited state of two sublattices, and Q is the coupling term. Our system is arranged into two sublattices, sublattice A and B, in terms of Rydberg population. In one sublattice, Rydberg population is higher, in another Rydberg population is lower. Sublattice A and B denoted by the red and blue color, which corresponds to 1 and 2. So for these two sublattices, we have get this four dynamical equation. And we can plot omega 1, omega 2, that is population imbalances for ground state and excited state for two sublattices with time for different values of on-site interaction, keeping other parameters fixed. And we get three different phases: uniform phase, antiferromagnetic phase, and oscillatory phase. In the uniform phase, omega 1 denoted by the red color and omega 2 denoted by the blue color are equal and constant over time. In antiferromagnetic phase, omega 1 and omega 2 are not equal but constant over time. And in oscillatory phase, omega 1 and omega 2 are oscillating with time. Now to characterize those three phases, we have defined an order parameter theta, which is the absolute value of omega 1 minus omega 2 by 2. And we have plotted theta as a function of on-site interaction u. And it is seen that when omega 1 equals to omega 2, we get theta equals to 0. This 0 theta line corresponds to the uniform phase. And this line corresponds the antiferromagnetic phase, and this fluctuating part corresponds the oscillatory phase. Slide is not changing. Sorry, এটা জ্বলছে মাউস দিয়ে না করেছে sorry ঠিক আছে Now to characterize these three phases, we have calculated the fixed points of the system and checked its stability. We can calculate the fixed point of the system with these conditions, and we get 3 branches of uniform fixed points and 2 branches of non-uniform fixed points. Here, corresponding to the uniform phase, we get one stable branch of uniform fixed point. And in the antiferromagnetic phase, we get 2 stable branch of non-uniform fixed points, and the branch of uniform fixed points become unstable here. In the oscillatory phase, we get here both the uniform and non-uniform fixed points are unstable. As there is no stable fixed point, theta is very fluctuating here. Now coming to the uniform phase, here we get 3 branches of uniform fixed points, among which only one is stable, another 2 are unstable, and 2 unstable branches of non-uniform fixed points. These are the parameters. Now we also calculate the correlation with semi-classical Monte Carlo simulation. To calculate the correlation, a configuration of the system must be constructed, and to construct a configuration, transition probability for 2-level system is needed. This is a picture of 2-level Rydberg system with many atoms per side, and this is the expression for the transition probability. Here delta delta modified has this expression. This is a reference for expression of transition probability of 2-level system. Now we have to construct a configuration. To do that, first we have plotted omega 1 and omega 2 as a function of u, and for a particular u we can get omega 1 and omega 2 for 2 sublattices, and from that omega 1 and omega 2 we can get rho e1 and rho e2. Here rho is 2 cross 2 density matrix for 2-level system, and rho e is Rydberg state population for sublattice 1, sublattice A and B denoted by 1 and 2. From this rho e1 and rho e2, we can arrange an array of lattice sites. We can construct an array of lattice sites by the help of Monte Carlo simulation. Here G denotes the ground state, R denotes the Rydberg excited state. But here we get 2 nearest neighbor Rydberg excited sites, which is not possible because we have nearest neighbor Rydberg blockade interaction. So with the help of this transition probability, we can rearrange the lattice sites where there are no nearest neighbor Rydberg excited sites. Now this is our configuration, and with this configuration we can calculate the correlation between I and I plus Rh site. Here AI denotes whether the ith site are in the ground state or Rydberg excited state. And we have plotted SR as a function of R for different phases by tuning U, keeping other parameters fixed. And it is seen that correlation is maximum in antiferromagnetic phase and minimum and nearest to 0 for uniform phase. In oscillatory phase, the nature of the correlation is same as that of in the antiferromagnetic phase, but magnitude is lower.
I'm afraid you have to continue right now.
Okay. This is a plot of SR versus R for 3 phases in a single picture. This red color denotes the antiferromagnetic phase, this blue color denotes the oscillatory phase, and this green color denotes the uniform phase. And it is clear from the picture that order of the magnitude of the correlation is different in 3 different phases. We have also plotted correlation as a function of on-site interaction, correlation between I and I plus 4th site, I and I plus 7th site, I and I plus 10 sites. And it is clear from the picture that correlation in antiferromagnetic phase denoted by the red color and in oscillatory phase denoted by the black color is positive for even side difference, that is S4, S10, and negative for odd side difference, that is S7. Here are the parameters. Now I can summarize our work. Tuning the on-site interaction, three phases can be found in terms of the population of ground state and the Rydberg excited state, uniform phase, antiferromagnetic phase, and oscillatory phase, and we have constructed an order parameter to characterize those phases. Studying the stability of the fixed points of the system, we can find the boundaries between uniform and non-uniform phases, and spatial correlation is studied by semi-classical Monte Carlo simulation. And it is seen that order of magnitude of the correlation is different in the three different phases. These are the references. Thank you.
Thank you, Ms. Suvechha Indu. We will be having a quick couple of questions.
Just 2 quick points to understand. Like the gamma is the dissipation coefficient. Just can we clarify what is this dissipation coefficient means?
Spontaneous emission.
And so then the next part will be the in the uniform antiferromagnetic and oscillatory phase, you mentioned the U values are 2, -0.3 and 0.6. What that actually signifies?
Actually, we take some particular values. Actually, we take some particular values of U. Actually, this is a range of U for which we get these phases. That is, in this range of U, we get uniform phase. In this range, we get antiferromagnetic phase, and in this range, we get oscillatory phase.
And what is that U actually denotes?
U is the on-site interaction. When U is negative, then there are attraction between the atoms in the ground state, and when U is positive, there are repulsion among the atoms or in the ground state.
The oscillatory phase, what kind of progress? What is the frequency of that oscillatory phase actually used?
Actually, we have not calculated
What is
this.
the frequency of the oscillatory phase? As I understand from this graph, it's really we find, but it's like not a uniform frequency.
No, no, no. This is not uniform.
Okay, thank you.
Thank you. Thank you.
Since we are running short of time, I would request all the audiences to have 2 questions at most, please. I'm sorry. Moving on further, I would like to invite Dr. Prasun Sarkar. He is currently a postdoc at IACS Kolkata. Over to you, sir.
Good evening, everyone. Firstly, I take the opportunity to thank the organizer for giving me an opportunity to present my work here, present my recent work. So today I'll be talking on this topic, tuning the lipid cycles with a noise, survival and collapse. So before I say, keeping it in mind that it's a mixed audience, just I want to say one thing about the lipid cycle. Lipid cycle is it's a very common thing in nonlinear dynamics. So basically, we need to have a self-excited system. So there will be damping, and there will be a self-excitation or oscillatory system, oscillations kind of things. Both these two will make a delicate balance to produce an isolated closed trajectory in the asymptotic limit, not in the short-term limit, in the asymptotic limit in phase space. So I have discussed it during my course of the lecture. So firstly, you give a brief motivation or introduction part, because the not it's a common fact that nonlinearity and stochasticity go on hand in hand and produce some interesting result in nonlinear. So, but the last couple of decades, so lots of noise-induced phenomena have been studied earlier. I won't go in details. One, and in some cases, in most of the cases, noise produce some positive results, which is counterintuitive to our expectation. So, lots of examples are there, like pattern formation, stochastic resonance, and all. But the thing is that, and it has been studied over a long period of time. So the thing is that main point is there, we make it unstable by the help of the noise, which acts on it by additive or in multiplicative form. So, but the problem is here. So it's a well-studied thing, but the problem is there that we usually it has in literature it has been said observed that it is being studied with the linear stability analysis, where the nonlinear equations is being studied near a fixed point only, and it is becoming linearized actually. But the problem is that when we move on to the nonlinear stochastic differential equation, it gives some serious problems because it cannot be closed. It cannot be make in a closed form, and linearization cannot be done here. Or mean field approximation also cannot be done here, because the thing is that since we will be studying this subject in the asymptotic limit, so it won't be able to find the generic dynamical behavior. The generic behavior will be lost if the equations of motions or that kind of things, like equations of nonlinear stochastic order. So, and in literature, no such study has been done in the analytical level. So it is worthwhile for us to explore the noise, how the noise affects the stability of a limit cycle in the asymptotic limit or the long time limit. So this is the point where we actually work here. And so a direct method is required, we thought. So the thing is that I have just summarized it in a usually in the linear models that's separated from the surroundings and non-distributive or deterministic. So these cases are easier to solve. We are in the realistic system, but the system is non-linear or coupled with surroundings, which creates dissipation and noise. This is a challenging task for us. And in the dissipation also, we have it can be either linear dissipation or non-linear dissipation. Earlier we have solved the linear dissipative case. So here we focus on the nonlinear dissipative part. So basic outline is that, so since we are trying to develop an analytical technique, so noise is too fast compared to the other time scales of the system. We use the separation of time scales and analyze it. The effect of the first motion will be subsumed into the slow dynamics, and the slow dynamics will be renormalized with the effects of noise, and it can be so. So, and from here, an additional interesting thing actually which we got, we will find a critical threshold where the limit cycle, we can find a condition that the limit cycle will be collapsed. So let's see how it goes. And we have tested for two types of limit cycle oscillator, Van der Pol and Duffing Van der Pol. So firstly, we take the generalized case. It's a self-sustained oscillator. So here is the nonlinear damping term or dissipation term, and it is being driven by an external noise. So since it is external noise, so no fluctuation dissipation relationship is required and valid here. And so, and C is a parameter actually. If fx is 0, so we get the, the potential is linear, so you get the standard Van der Pol, and if it is a non-zero cubic type, so you get the Duffing Van der Pol. Let us test our theory. So this kind of correlation actually the noise follow. It's a white noise, delta correlated. So the analytical technique actually we separate it in a slow variable and in the fast variable. The capital omega is the inverse of tau c, that is the relaxation time of the noise. Ideally infinity, but it is a large one and much larger than any other frequency or something like that in the system. So if we just substitute it, so we can separate it out the dynamics in a slow part and a fast part. Okay. The interesting thing is that the slow part, here we get the size square term, which is been subsumed in that slow dynamics. The fast motion, so noise effect is being noise has been affected here, and the fast motion is something like that, which is very much complicated and cannot be solved analytically. So the thing is that since we have taken the approximation that omega is much larger than any other frequency, so we can drop, so psi double dot is of the order of omega square. So it's a different time scale. So we can neglect, so psi double dot is much faster than any other, all the other things in that equation apart from the stochastic part. So we remove all these terms and find an approximate solution for the first part, psi double dot. And this kind of equation actually arises in heating of plasma in electric field. We got the reference in Van Kampen and some literature by paper by Sarkar and Hall. So from here we solve it with the help of noise and other, and find psi average is 0, but psi square average is non-zero. It is being introduced here in the slow dynamics. So the another interesting thing is that the first slow part now is a purely non-linear dynamical equation, and the first part is a stochastic one. No deterministic part is concerned here. So we find with the help of this one, we find psi square, and now we want to. So since it is renormalized now, so we need to solve the slow variable x, and it can be solved with the renormalization group method itself that was developed earlier by Goldenfeld and Uno, followed by Biswas also. So, but here actually we use the Lindstedt-Poincare method and just expand it in terms of this one. And for brevity, we will skip the mathematics actually. So we get order by order equations, and the frequency and will get dressed or the modified. So these are the up to second order equations in the earlier slide, and we find out the 0th order and first order equations and solution with some initial conditions. So from here we find the first order frequency correction is 0, but we get a with the circular term divergence, removal of circular term divergence, we find the amplitude of the limit cycle, which is the order of 2 alpha. If it was purely a deterministic one, so alpha would be 1. So it's a only 2. The radius of the closed trajectory will be 2. It's standard and has been reported earlier also in literature. So, but the thing is that now we have modified it, the amplitude of the radius of the limit cycle with the noise, and so it has been modified, and now we find the moving further first order and second order correction. So the frequency has been raised by something like that, and the final solution for xt or the small x average is something like that. So these are the basic parameters. Now we want to see the figure or the shape of the limit cycles. What will it be? So firstly, we have plotted it which is in the phase space in the asymptotic limit. All the figures actually I have shown here. It has been considered for at least 1,000 trajectories, and the time is after 10,000 units. Otherwise, we won't get the asymptotic behavior. So the actually, as I have said earlier, whenever d is equal to 0, d is the noise strength. It has been absorbed in alpha. When it is d is equal to 0, it's perfectly in the deterministic limit, which has been this is a standard textbook problem. So we have just reproduced it. Now, if we keep on increasing d further from d is equal to 0.5, so now the radius of the shape of the limit cycle gets tuned with the noise. As long as, so if we keep on increasing the noise strength, it gets reduced, and in each cases we have compared it with the exact numerics, which is the equation number 1, which, without any taking approximations and all. So not only that, and if we keep on increasing,
Sorry
we
to interrupt you.
sorry.
You have 2 more minutes.
So it has been the set has been earlier more reduced. And not only that, as I have told earlier, so we get a critical condition. From that condition, alpha square is equal to 1 minus this equation. When psi square greater than 1, it should collapse. The limit cycle should collapse. So theoretically, we get this kind of critical value, and we have plotted this one for the one is theoretical and one is numerical. So that time the limit cycle completely collapses. So this is the basic phenomenology we have mentioned there. And for the testability of our scenario, actually, we have taken another the workability of the Paget stream. We also extend it to another kind of nonlinear differential equation having self-constant oscillator. That is Duffing van der Pol. Here the difference is that apart from the nonlinearity in damping or dissipation, there is a nonlinear term in the potential also, which makes the situation much more complicated. And still we get the proceeding in the same way, but the mathematics is little bit difficult compared to this one. We get the same kind of results, and the frequency gets 2 times modified, one due to noise and one due to dynamical correction. This is for noise and this is for dynamical correction. So that's all. So again, we are getting the similar kind of results. Here also we have taken the critical value around 8.17. So at that point of view, we have taken the theoretical and numerical plots, the limit cycle collapses here. So in summary, just in a minute I am finishing it. So as we have proposed a analytical technique for the nonlinear stochastic dissipative system, which is oscillating. And with the help of separation of time scale, we have solved it. So as I have mentioned earlier, it's a limit cycle is a delicate balance between the damping and the self-excitation, resulting in an isolated closed asymptotic trajectories in the phase space. So linearization won't give you the picture at all. So it's not a good option. And we have taken the technique which is used in vibrational mechanics to the stochastic dynamics. And as we have already said, the thing is that our future plan is that it has been done for white noise. Now we can extend it to the colored noise system having finite correlation time, and also for the system which have multiple limit cycle times. And also another what I have already submitted, that is actually we are extending it to the quantum limit or in semi-classical domain. But the noise plays the role here, that actually there the quantum fluctuation will play the same role. Whether it is going in the right way or wrong, we have tested it, but it is going. It has, I will talk it in later. So that's all. So this is for detailed reference. And I must acknowledge my supervisor, Professor Dev Shankar Rai. And of course, obviously, Professor Janto JKB sir, he actually gave instinct that it may happen. And my junior actually, who is in a postdoc at Tennessee. So thank you all.
Thank you, Dr. Prasun. We'll be having a quick couple of questions.
If we understood correctly, by using the noise, what you calculate, the change in the omega zeros and all, that could not be done without noise. Meaning the original limit cycle, you already knew the omega zero and all that, right?
But the thing is that with the help of noise as an external perturbation, that also affects the frequency or the event in the damping. Since those are in nonlinear, so it brings some unpredictability in the system. How the dissipation or the potential is being changed with the in presence of noise, that is our main interest. And whether it affects the limit cycle or not.
How the potential get denormalized.
Even the damping also gets denormalized.
The very first beginning you mentioned separation of time scales leads to fast and slow dynamics. How is this separation of time scales being followed?
Followed, yeah. If we, suppose if we go for this one, this equation actually. So this is that entire equation. So Zt is the noise, which is very fast compared to any other time scale of the system. Okay, so I can say, so I can see it, I can view it. So now the technique is that how can we deal it mathematically? So one technique is that direct you separate it directly. This is a slow part and this is the fast part. Okay, so this kind of yeah. Yeah. So this technique actually used in vibrational mechanics, originally introduced by Blackman. So it's another BPS, Blackman perturbation scheme. So usually is applied to deterministic system. We have, for the first time, we have extended it to the stochastic dynamics also.
Why is it only slow? Why is the hybrid cases there is no kind of hybrid case?
No, vibrates actually, in case of vibrational mechanics, oh, slow and fast. Oh. No. So that is yeah, I got your point. Luckily, there is no such time scale, which is in intermediate between the slow and fast. If it is there, so definitely the things will be much more complicated. We are we're trying to solve that kind of right now. If there are more than 2 or 3 time scales, how to solve them, treat them analytically. If you solve it numerically, then it is very easy. That is the other thing. But we have to solve it analytically so that we can understand what is going on inside the problem. That is our interest. And then definitely we will verify with the numerics. Otherwise, nothing will stand. Yeah. Capital omega, yeah, that is 1 by tau c. Tau c is the correlation time of the noise. So ideally tau c is 0. So omega is ideally infinity. Yeah. So that's why tau c very small. So that omega is 1 by tau c is very large. So from here we can easily separate out the time scale. Whenever the capital omega is there, so the time scale is there. Yeah, exactly. It is correlated, but with a finite, very small time. Thank you.
Thank you, Dr. Prasun Sarkar. Now, it's time to welcome the last speaker of the session, Dr. Hillol Biswas. He has done his PhD research from National University of Singapore. He is currently working as a postdoctoral research associate at Harish Chandra Research Institute. Over to you, sir.
Hello everyone, I am Hillol, and today I'll talk about something called time crystal in a driven central spin model. So first, let us take some time to understand what time crystal is. Spontaneous symmetry breaking is a very big theme in modern physics, and more often than not, phases of matter is classified by the symmetry it breaks. For example, if you break continuous translation symmetry, you will get crystal lattice. So one such similar, very profound symmetry is time translation symmetry. So it says that if you have a Hamiltonian that is time invariant, you will get the ground state which is also time invariant. So in 2012, Frank Wilczek raised a question that can we break the continuous time translation symmetry? So that means with even with an time invariant Hamiltonian, you will get the ground state to be moving with time. And in analogy with space crystal, they called this kind of system time crystals. But this sounds perilously close to something like a perpetual motion machine. But and very quickly people showed that in equilibrium you cannot have a ground state or any excited state that is time dependent if the Hamiltonian is time invariant. But the question still remains that can we have time transition breaking symmetry breaking if in non-equilibrium setting? And this has been a very fertile area of work, and lots of people have shown time crystal in various settings, and the most widely studied of them is this quantum systems with periodic drive. Let's take an example. Suppose we have a bunch of spins all pointed in upward direction, and you apply a perfect pi pulse. So it will every spin will rotate by 180 degree and come to downward direction. And if you apply another pi pulse, it will go back to the original state. So the Hamiltonian is like Hamiltonian is time, not time invariant, so you don't have continuous time translation symmetry. But if you look at it stroboscopically at time t, 2t, like that, the value of the Hamiltonian is still same. So it has a discrete time translation symmetry. And when you look at an observable, like here, if you look at the magnetization, you will see it comes back to the original state after period 2t. So we have Hamiltonian that is of time period t, but the observables have time period 2t. So you are breaking discrete time translation symmetry. But here, this is predicated on the fact that the pi pulse have to be perfect. If there is any error, it will not come back to the original state. So if we want more than a point in parameter space where we can see time crystals, we'll have to add certain interactions to the system. And this is where this one example of this is central spin system, where we have a central spin, and there are a bunch of satellite spins, all are connected with this central spin with the same strength. And this is the interaction Hamiltonian. SI are the individual satellite spin, and sigma is the central spin, and this is the interaction part, and this is the drive part of the Hamiltonian. You can see that we can there is a provision of driving the central spin and the satellite spins individually, and these will need later. So this is the time evolution factor concerned with one kick, and if we expand one term, we will see that if we kick one spin just once, it will become a combination of the flipped state and the original state with certain with appropriate coefficients. So let us consider that we have all up spins. So this is the satellites part of the wave function, n up spins and 0 down spins. Along with that, we have up state of central spin also. So if we give one kick, if we drive the system once, so we'll have this all combination of all up spins, all down spins, and everything in between with appropriate coefficients according to this expansion. And along with that, we'll have the cos and sine component of the central spin also. Now, if we give it a second kick, so there are two phase factors coming in. One is the geometric phase coming in from the two kicks, and we have the dynamic phase factor that is accrued during the time of two cycles, and they will compensate each other to come back to the original state. So here you can see that we have calculated for 20 spins. We're starting with all up spins. After every two click, it will come back to the original state. So there is a period doubling time crystal here, exact period doubling time crystal, and this happens for any value of this driving factors. So if there is any error in the pulse that you are providing, it doesn't matter. It will still come back to the original state, and it doesn't only happen for all upspin state. If we start with any random state, suppose here we have started with 16 upspins and 4 downspins, so we have a net magnetization of 12, and here we also are saying that it will come back to the original state after every 2 kicks. So we are breaking the time discrete time translation symmetry here. Now let us take a look at it analytically, how this compensation of phase happens. So the analytical calculation we can only do at lambda equal to pi by 2. Otherwise, we have to do numerics only. So at lambda equal to pi by 2, let us look at, let us try to calculate the probability that this central spin will come back to its original state. So after one kick, we'll have this combination of states, and let us call it psi satellite. And along with that, we have these two components flipped up the central spin and the original state of the central spin with a certain appropriate coefficients. So if after the second kick we are trying to find the probability of the central spin remaining in coming back to its original upspin state. So this one has to remain in upspin state. This cannot flip. So it will, after the second kick, it will get another cos in its coefficient, and this part will have to flip. So it will gather another I sign as coefficient. To its original state, but these three are for 20 spins, and we can see that it is following the drive, and it is following only GC. So in these two, we have changed the value of GS, the driving potential or driving strength of satellite spin, and it doesn't matter. Okay, it doesn't matter whether the strength of the satellite spin doesn't matter, and if we change the driving strength of the central spin, and so if we increase GC, it varies faster. Now we have calculated a lambda dependent phase diagram at certain value of GC and GS. So this is lambda equal to 0 part, which is essentially non-interacting, and lambda equal to pi also, if you see this expression, it will give you the same value. It will give you always 1, so it is equivalent to lambda equal to 0. So these two are non-interacting phases, and at lambda equal to pi by 2, we have this interacting phase. And these non-interacting and interacting phases are separated by a very sharp time crystal phase at lambda equal to pi by 4 and 3 pi by 4. Here, what happens is we end up with a longer period time crystal. So at lambda equal to pi by 4, here also we see the even and odd parity. For the n equal to 20, we see that this is a satellite spin curve, and this is a central spin curve. So they follow the same pattern, and they have a 12 period time crystal. But for for odd number of spins, they follow different patterns. We don't yet know how. We are still working on it, but we have a 24 net 24 period oscillations. So we don't have the analytics for this. We are trying to do some more calculation to understand what is happening here. So the summary is this, that we have this exact period doubling time crystal for any value of driving strength, and at lambda equal to 2 for any value of driving strength, and any initial state, and at lambda equal to 2, we have an analytical explanation for the period doubling time crystal and for the odd even parity. And there is, so this analytical explanation hasn't been done by anyone else till now. And this longer period oscillation at lambda equal to pi by 4, this sharp time crystal phase, that also hasn't been seen by anyone till now. So these two are the new results that we are seeing. So let's open it up to questions if anyone has. Yeah.
Any questions?
So what is the origin of this geometric phase here?
So the origin of the drive. Yes, this is a so I probably shouldn't have called it geometric phase. It's a phase.
Dynamic phase, I understand because of the drive.
Yeah, no. The dynamic phase is because of the evolution of the state, right? So the drive is at exactly at this drive is the delta function of time. So it is happening at spacious at this exact moment. But between two drives, the state is evolving. So that is the dynamic evolution of the phase, and the extra phase that it gains at exact nt times, that is what we are calling the geometric phase. So these two are the phases that are compensating each other. Time evolution during the two kicks, the dynamic phase accumulation during the two kicks, and the phase we are providing it by driving it at exactly nt moments. These are the two phases. Yeah, I shouldn't have called it geometric phase. So this is phase gathered due to drive and phase accumulated due to the dynamic evolution of the state. These two phases.
Another question is that, like in the last many slides, you have shown this plot. What is the x-axis and y-axis?
So the x-axis is
Not this one.
yeah. So the x-axis is time. So this is the number of kicks here also, and here this is the number of kicks, and y-axis is the magnetization. So the probability that it will come back to the original upspin state. So if the probability is 1, it will come back to fully upspin state. Magnetization will be 1, and if it doesn't come back to the original state fully, so magnetization will be less. And here it is -1. So it follows this cost term. It varies from +1 to -1 and back.
Yeah, very nice question. You say that the doesn't have specific time.
It doesn't have continuous time.
Then the state should not be time dependent.
No, so it has this discrete time transition symmetry, because at every nt you.
Example. Doubt is the following. Suppose I have a simple harmonic oscillator.
Okay.
It doesn't have any explicit time, but the state is oscillating time between the state.
The simple harmonic oscillator. Yeah, so but that is evolution of the evolution of the dynamic phase that you are getting by applying time evolution operator. You get the dynamic phase evolution. That is there for all kinds of state, right? But it doesn't change the value of the observable. If you apply some operator and calculate the observable, you will have e to the power minus iht or into matrix into the plus iht.
Quantum oscillator, if I calculate the x square average, where the particle is, that will time because the particle is oscillating. X average, let's see. Quantum x average.
Okay.
I'm going wrong somewhere.
Yeah. So this is like you are so quantum oscillator is like you are leaving the particle someplace and it just spreads. Yes, exactly. It spreads. So this is it is a transient state. It's not an equilibrium state, right? If it's still it is still moving.
That is the special part. The quantum oscillator still shows oscillation, right? In x average. Then it is constant, but
I have
Station is fifteen. If the station is 15, there is no time dependence.
x average is zero oscillation.
But the happy station is the all those amide polynomials, and then I mean, if I
With the definite parity, each state has definite parity. So the examinability will be zero.
oh sorry.
Thank you, Dr. Hillol. With this, we come to an end of our second oral presentation session. I will be handing over the mic to Shrabanti.
Thank you, Nandini. Now we will have a brief discussion, brief session with Wiley. And I would like to ask Seheli Haldar, a PG2 student of Presidency University, to conduct this session. Seheli.
এটা পড়ে যাচ্ছে